Hello everyone, and I would like to begin by thanking the organizers for this wonderful opportunity. In the next 10 minutes or so, I would like to show or present data which shows that IRF3 regulates inflammation in different neuroinflammatory conditions. So, oops, I cannot move. Okay. Pathological interferon signaling has been observed in variety of different neurological disorders, such as Alzheimer's disease, Parkinson's, traumatic brain injury, and even aging. Um, in addition, there are interferon responsive cell types, including microglia, astrocytes, and oligodendrocytes that have been observed in these disorders. And at the heart of interferon signaling, we know that there are interferon transcription factors. There are 9 of these, and out of which, for my today's talk, I will focus on interferon response factor 3. So, IRF3 is no stranger to innate or adaptive immunity. Its role downstream of TLR3 or TLR4 is very well studied in the peripheral models of either viral infections or bacterial infections or sterile inflammatory conditions such as alcohol abuse or obesity. While most of the studies in the central nervous system are restricted to viral encephalitis. Now, IRF3 gets indirectly implicated in a lot of neurological disorders through the engagement of C-gasting pathway, and we heard a lot about that earlier today. However, very few studies have directly investigated the impact of manipulation of IRF3 on the features of these neurological disorders or the interferon responsive cell types that are identified within them. And therefore, we set out to study effect of IRF3 in different neuroinflammatory milieu. We began by studying the most commonly used model of neuroinflammation, which is LPS challenge. Here we administered LPS to wild type and global IRF3 knockout animals for about 6 hours. And one of the most striking feature was the reduction in or attenuated sickness behavior. So the wild type mice, when administered with LPS, showed reduced exploratory activity in the open field test, and there was a significant rescue of this phenotype in IRF3 knockout animals treated with LPS. Next, we also investigated various pro-inflammatory transcripts from microglia and astrocytes, which were isolated from the brain tissue of these acutely treated animals. And what we observed that interferon response genes, which were induced in the wild type LPS condition, as expected, showed great attenuated response. But in addition to that, genes, other pro-inflammatory genes such as COX2 or members of MHC class 1, or for example, GFAP in case of astrocytes, again, which were induced with in the wild type animals with LPS treatment, showed very dampened response in the IRF3 knockout animals, suggesting that the anti-pro-inflammatory effect of IRF3 deletion was not just limited to the direct interferon responsive genes. Next, we also looked at the levels of cortical cytokines, and there was marked attenuation of the secretion of members of interleukin family. In addition, there was reduction in the release of chemokines such as MCP1 or CXCL. And now that we saw reduced release of IL-1 beta, we further wanted to investigate the inflammasome pathway, which is responsible for the cleavage of IL-1 beta. And for this purpose, we used a slightly different model of LPS, where LPS was administered for 4 days to the wild type animals. And here we could reliably pick up activation of inflammasomes in the hippocampi of the wild type animals. However, this response was strongly attenuated in the IRF3 knockout animals. Interestingly, in addition to reduced inflammasome activation, we also observed that the infiltration of myeloid cells in the brains of these animals was reduced. For example, with repeated alcohol doses, we saw that there was increased infiltration of myeloid population in wild type animals, and there was almost complete inhibition of this phenotype in the IRF3 knockout mice. So, to summarize this part, we were able to show that IRF3 deletion showed protective effect against a variety of pro-inflammatory mediators of LPS, such as cytokine production, sickness behavior, myeloid cell infiltration, and even inflammasome activation. We also validated direct role of IRF3 downstream of TLR4 signaling in microglial cultures. Now, next, to investigate the effect of IRF3 activation specifically in microglia, we took advantage of IRF3 2D animals. Here, IRF3 is rendered active by it's rendered constitutively active by phosphorylation of two key residues. And we drove the expression specifically in microglia using CX3CR1 CRIA driver. We confirmed the expression of IRF3 by presence of a doublet of IRF3 in the westerns done on the flow sorted YFP positive population. And what we observed was mere expression of the active form of IRF3 was enough to drive a very reactive or a pro-inflammatory morphology of these microglia, as seen here by filament tracing, which is less branched and thicker filaments. And it was associated with a highly pro inflammatory transcript. But what was the most striking feature was that in our volcano plot, some that stood out were the genes which were associated with AD. Our top candidate was ApoA gene, and we've been able to validate its upregulation at protein level as well. So, to summarize this part, we were able to show that mere activation of or mere expression of IRF, active form of IRF3 in microglia in the absence of any additional stimulus drove a very strong pro-inflammatory phenotype in microglia, which was associated with, again, myeloid cell infiltration. We also observed cortical astrocyte proliferation and importantly, upregulation of genes associated with AD. And this phenotype really allowed us to, or really prompted us to look at how the transcriptome of microglia in a mouse model of AD would appear if we deleted IRF3. And therefore, we performed bulk RNA sequencing on microglia that were isolated from 5xFAD model of Alzheimer's disease. We used 6-month-old mice and compared the transcriptome of these microglia to that of a double transgenic IRF3 knockout 5xFAD animals. And we observed that there was marked reduction in the interferon signatures as well as antigen presentation in these microglia. We have also validated some of the members involved in either antigen presentation, also AXL, which is an immediate early gene following interferon response, and observed that the expression levels were significantly downregulated in the IRF3 knockout 5xFAD background. Moving on, we wanted to test the effect of IRF3 deletion on other features of AD. And so we looked at amyloid pathology in these animals. Although we have not seen a significant effect, there is a strong trend towards increased plaque pathology or plaque count in the cortices of these animals. And we are currently a little unsure about the mechanism here, and we are investigating different other components of or features of AD, such as synaptic engulfment by microglia or the tau load or neurofilament activation, phosphorylation, I'm sorry, in this model. So to summarize the last part, we were able to study the effect of IRF3 deletion in the Alzheimer's disease background, and we observed that IRF3 deletion led to a marked attenuation of the interferon signatures in microglia. And I forgot to mention that this reduction in interferon signatures was accompanied by relatively no change in the DAM genes. So basically, this would allow us to study effect of IRM population, if anything, in isolation without affecting the other disease-associated microglia populations. So, taken together, we were able to show that expression of IRF3 in microglia is important in different neuroinflammatory responses. And with this, I'm happy to take questions.
So we have 2 questions we can take up here. Then one here.
Thanks for the presentation. You built up a nice story with the LPS model leading into your KO model, and then I was expecting something else in the AD model. Interestingly, you don't show anything related to the LPS or anything related to the knockout model. Can you comment on the impact on infiltrating leukocytes in your AD model and what's happening with the astrocytes in your AD model? Thanks.
Yeah. So we have isolated astrocytes, and we are still investigating the effect on the pro-inflammatory features of astrocytes. So I do not have data to that end as of now. Or coming back to the effect of infiltration. So most of, I have looked at mainly the myeloid infiltration through flow cytometry so far, and I am not able to pick up any significant infiltration in my model, at least in my hands in terms of AD. But it would be interesting definitely to look at T-cell infiltration or either if there is a low level of infiltration, which is probably not being picked up through flow, we are planning on doing stainings to see if I can pick up some signal over there.
That was a really fantastic talk. There's lots of interesting strands to follow up with that.
I don't know if she hears you.
She can't. Can you hear me?
Yeah.
Sorry. Quite loud anyway. Okay. So my question relates to the IRF3 knockout crossed with the Alzheimer's mouse model. Do you see the microglia forming the barrier effects around the plaques? I think that would be an interesting thing to look at.
Yes, so largely they do. I haven't systematically quantified that yet. But looking at the images, it looks like the barrier effect is intact in the IRF3 knockout animals.
Okay, then thank you very much. We have to move on. And we have like, now, applaud, sorry.